Hello everyone, my name is Ifrah Sajjad. I'm a second-year student attending Rutgers University, majoring in computer science. I've spent the last few months researching under Dr. Vikas Nanda's guidance in his lab, researching elastic proteins through the utilization of computational analysis. Elastic proteins are characterized through their unique structure and their abnormal amino acid sequences. Unlike normal proteins, elastic proteins have high resilience and the ability to deform to large strains with little force. The elasticity of elastic proteins is caused by their specific proline and glycine concentration. Since elastin is a more well-known form of elastic proteins, we can analyze its backbone and solvation entropy as an indicating factor when searching for more types of elastic proteins. By understanding the characteristics and structure of the elastic protein, elastin, we can look further into proteins with similar characteristics to help us find more types of elastic proteins. Through the use of computational analysis, we are searching for other proteins similar to elastin. Unlike normal proteins, which become deformed and misshapen when stretched, elastic proteins have the ability to return back to their original shape. Due to the lack of research on elastic proteins, we hope to find more through analyzing elastin structure and sequence. Elastin is the main indicating factor in our research. Elastin consists of a high concentration of amino acids such as proline and glycine. It also has a high backbone entropy, which enables the proteins to be flexible without damaging its original state. My portion of the project was to research and test different clustering algorithms on the human proteome sequences found on UniProt, a protein database. I tested these algorithms utilizing the coding platform Jupyter Notebook, coding in the language Python. I utilized matplotlib to create various graphs and scatter plots for my code. I utilized sklearn's library for the clustering algorithm as well. Heading into my results, Figure 1 depicts the backbone entropy versus the water entropy of the entire human database. The red and purple regions represent two initialized clusters from the k-means clustering. Figure 2 represents the same graph except with 4 initialized clusters instead of 2. K-means is one of the many clustering algorithms that was tested and researched prior to creating the algorithm that we are currently using. The way K-means works is that we first choose the number of clusters K that we want to place the data into. Then, we randomly set K points, otherwise known as centroids. Once this is done, we identify the data points closest to each centroid. This is done by calculating the distance away from each data point to each centroid. Then, we assign the data points to each centroid, and once this is done, we then calculate the mean of the points in each cluster and move each centroid to that mean point. Utilizing this algorithm will furthermore allow us to automate our search for elastic proteins rather than manually, individually observe each protein. And looking at figure 3 in the upper right-hand corner, this graph depicts a density plot of what we presume the clustering algorithm would return. The dark blue concentrate in the middle is where we assume the clustering algorithm would create a cluster since the middle region is where more proteins lie on the graph. And elastic proteins on this graph are going to be the tail towards the bottom right of the graph where the circle is depicted. And although the algorithm was able to cluster our data, it didn't do so in the way we had hoped. For the future, we hope to test other organisms' proteomes, and we also hope to test other amino acids for their frequency in the human proteome, as well as possibly try all 12 types of clustering algorithms and see which one returns a better result. Thank you.